So let's go. We are 2 minutes past.
Okay.
Welcome everyone, and thank you for attending this new webinar by the Global Plant Council. This time we have with us the Professor Ros Gleadow from Monash University in Australia, who will be talking about the nutritional quality of cassava in a warming world. She is the spokesperson for the International Working Group for Safe Cassava. And just as a reminder, this webinar will be recorded and streamed through Zoom and YouTube. So keep that in mind. Uh, Questions will be reserved for the later part of this webinar. You can use the Q&A tool that it's available on Zoom, and we will answer your questions by the end of the talk. But first, a few words about the Global Plant Council. You may already know that the Global Plant Council is a global coalition of plant science organizations and societies that it's with the aim to raise awareness of the importance of plant science for society and building a community for plant scientists and represent them whenever necessary. We are present online on almost all existing social media platform, and you can reach us using those means. A little bit about what we do. Our main points of action are showcasing the latest plant research, and that could mean several things. One is highlighting the newest research on our news section and blog section in the website, but also organizing things like this one, webinars and other activities also in person, like workshops and conferences that we also attend. There is another big part of our work. It's related to the Early Career Researcher International Network, sorry. That is a collection of activities addressed to help early career researchers in plant science on 4 essential pillars that are the job hunting, grant writing, and improving their communication and networking skills. And last but not least, we also do policy and advocacy work for the plant science community. You can see that in our statements that are policy statements that are available on our and highlighted in our website, but also in other kinds of publications that are usually published through our blog and social networks, where we advocate for the scientific societies and their importance for organizing scientific work and scientific interest around the world, and also by participating in different policy global arenas, such as the UN conferences for climate change and the UN conferences for biodiversity on behalf of the plant science community. All our activities are put together and organized on our website that I invite you to visit. We also host lots of resources of different kinds that could be useful for the plant science community and also plant science adjacent communities such as farmers, educators, and others, some policymakers. So thank you on my behalf, and you can always reach us through our emails and website and social media, as you know. And now I will leave the floor for Ros to talk about cassava and how climate change might influence how cassava um, is resulted the fruits. So Ros,
That's good.
floor is yours.
So
Ros is
thank you.
old old acquainted of the Global Plant Council. I will say a couple of things also about her on the chat.
That's good. Thank you, Isabel. And as Isabel said, we got to know each other when she's the Chief Communications Officer for the Global Plant Council, and I was president for about, 4 years and have had various other roles. So we really appreciate the Global Plant Council hosting these for us. And I do notice that people are putting in the chat where they're from. I think this is fantastic. And if any of you, your organisations or your universities would like to be part of the Global Plant Council, please contact Isabel at the Global Plant Council. Perhaps you could put your email address in the chat there, Isabel. The cost is very much dependent on your ability to pay. So if you don't, if you come from an organisation that doesn't have very much money, I'm sure it could be negotiated what you could get, and then you can get extra access to different things if you're a member. But most of what the Global Plant Council does is freely available throughout the world. It is something that they give to the world. So welcome everybody, and I'd just like to now, I'll just share my screen. So I become very small. Do I need to hide this? Hide video panel. There we go. No, hide floating. There we go. So today I'm going to so I'm just going to get out of that because that's all just gone doesn't work. So I'll just, for some reason it went straight to my next slide. I've been having problems with my computer.
Now it is working.
Now it is working. So if I hide these meeting panels, I think I need to hide them. So I'm going to talk about this nutritional quality of cassava in a warming world. I'm very aware that most of you who are listening to this seminar know far more about cassava than I do. I come from Melbourne in Australia, which is not a tropical area, and you can't grow cassava outside. So my experiments have all been done in greenhouses. You notice on my address, I've also called it Naarm because that is the name for the area where I live of the First Peoples of the area. And just before I start, according to tradition in Australia, I want to acknowledge the traditional owners of the land, the Wurundjeri people from the area of Naarm. So in my research, I started off, where did I come from? I started off trying to answer the question, how do plants partition resources at elevated carbon dioxide? And I started working on these particular eucalypts. They're called sugar gums. Most eucalypts are not cyanogenic, which means they contain these cyanide compounds. And this was just a vehicle, an interesting sort of plant to work on. And if you look on the right here, if you've got a certain amount of resources available to the plant, they can either be divided up between, say, photosynthesis, which leads to growth, or defense. So it might be some toxic compound or very thick, tough leaves or something like that. And that makes them longer. So there's different strategies for survival. And I've done all my research really on looking at how all these different things like temperature, herbivory, rainfall, and so on, and changing carbon dioxide affect this partitioning. And I use sugar gums because they contain compounds called cyanogenic glucosides, which are the same group of compounds that you get in cassava. So the rationale is that if there's something like a drought or very high temperatures that stops photosynthesis, plants will reallocate their resources to defense and they may become more toxic, for example. On the other hand, if for some reason you could stop the defensive strategy, you might get more photosynthesis. And more latterly, I'm not going to go into today, where there is a bit of crosstalk between all of this, which makes removing these compounds a little bit more interesting. So about 5% of all plants are cyanogenic, and an even higher proportion of crop species. And so some of the ones you might be familiar with besides cassava, which most of you will be familiar with, are like the apple seeds. You can smell that marzipan in there, which is made from bitter almonds, the apricot kernels. There's about 30 different types of eucalypts that contain these compounds. So what I'm going to talk about today is a little bit about cassava as a plant, about cyanogens and how you make safe cassava, but mainly we're talking about the environmental factors and then introducing the network that you're all part of. So as I'm sure you're very aware, cassava is an absolutely brilliant crop. It yields very high. You can grow it on poor soils. It can tolerate high temperatures and rainfall. And so we know that it's a very important food security crop, and it's also a very important cash crop. And in Southeast Asia is primarily grown as a source for industrial starch. So there are huge, it's grown all over the place. Its origins were in South America. I'm sure you're aware. It was taken initially by the Portuguese to Africa, where it is now very, very widely planted, and more recently in the last 100 years into the Pacific Islands. 1 billion people eat cassava every day. Here it is, manaho or manahot esculenta, whether you call it cassava, manioc, manioca, yucca, different names. A lot of people eat it, and of course, most of those 1 billion people don't have any problem at all. It's just a completely safe crop, but you've got to know what you're eating. And the peel can be very toxic. And for example, this is now for sale in Australia, and the average Australian would have absolutely no idea that you shouldn't eat the peel. So I've used this word already without introducing it. It's cyanogenic. That means it contains cyanogenic glucosides. And there's two different ones in cassava. There's linamarin, which is synthesized from the amino acid valine, and there's lotustralin that's synthesized from isoleucine. It's the same two cyanogenic glucosides. They're actually found in lima beans and trifolium, lotus, and flax. So it's quite well understood how these are made. The synthesis in cassava is mostly in the leaf tips, and it's then transported and stored in the roots. Though there is Kirsten Jurgensen from Copenhagen has very clearly shown that there is additional synthesis in the periderm of the roots. So, some people have actually thought about, could you interrupt this transport and so on, but I'm not going to talk about that today. So here's the biosynthesis. I thought I'd take you through this stuff. You may or may not know it, but you have valine or isoleucine. There's three enzymes. They're cytochrome P450s. There's two at the first level. These are alternatives. There's one at the second level, and the oxime is an intermediate, which is very toxic and needs to be quickly converted into this hydroxynitrile. That is unstable in that form, and it's stabilized by a sugar being added to it, and that will, depending on whether this has as its source valine or isoleucine, you get linamarin or lotustralin. So that's the pathway. Now, why are that most cyanogenic plants only have one enzyme at this first point, one of these CYP79s? But it's understood now for some work that Jess Lyons and Bredesen did in the Nature Biotech paper, is that that there has been a whole genome duplication at some point from an ancient paleo tetraploidy, and that these are paralogs, and both of them change valine to lotustralin, and both of them do isoleucine to lotustralin. So they don't, not one does them, one does the other. It's a redundant pathway. Well, not completely redundant, as we'll see. The enzymes form a metabolon. This has been demonstrated for sorghum, but not for cassava. And the genes are clustered on a single chromosome, so they're inherited together as a block. The reason I've bold the CYP79D2 is that this seems to be the most important of these two enzymes. Now, the cyanogenic glucosides, it's called a glucoside if it's got a glucose on it. It's a glycoside if it's not a glucose. That this is the basic pathway. Here's the little cyanide group with its triple bond, which is the thing that's very reactive and very toxic, stable with a sugar and a couple of amino groups. That's the generic one for all 60 or 70 different types of cyanogenic glucosides. There's a special enzyme, a beta-glucosidase, that breaks the sugar off. When that breaks off, it reverts back to that hydroxynitrile, which is unstable, and it can either spontaneously release an aldehyde or a ketone, depending on the glucoside. In this case, for cassava, it makes aldehyde and releases free cyanide. Free cyanide is toxic because it binds to cytochrome C oxidase in the mitochondria and causes death of herbivores. So the plants would kill itself if this was just this enzyme was here mixed up together in the cytoplasm. So what they do is that there is either tissue localization, they're in different tissues, or they're in different organelles, or in sub cytoplasmic compartments. So it's only when that's crushed, brought together, chewed up, mushed in some way that you release the cyanide. If you don't have beta-glucosidase, you don't release that. On the other hand, it can be broken down by sort of very strong acids and so on. So and by a lot of microorganisms. So generally, it's going to be toxic if you consume it. Now, of course, you can detoxify it, and I know Torkild and all these other people are online who are neurologists, medical people, and you know far more about all this than I do. But you can detoxify cyanide by combining it with sulfur, which comes from amino acids, to make a thiosulfate. It's catalyzed by an enzyme called rhodinase to make a sulfite and a thiocyanate, which can be excreted in the urine. And the sulfur-based amino acids are methionine and cysteine. And interestingly, the leaves of cassava actually are particularly low in methionine. So having a source of sulfur amino acids, methionine and cysteine, particularly you need to have some sort of protein other than the cassava leaves to really be able to do this efficiently. Again, far more people on this Zoom meeting than I am as an expert on this. But if you eat a monotonous diet that's high in cyanide, you can have, like these children have on the right, konzo, which is really why we're all here. These children have various stages of konzo from eating poorly processed cassava. This is a picture from Nasualo and Cliff in the PLOS Neglected Tropical Diseases issue, which is an excellent article if you want to read about it. But if you can have acute poisoning, which will depend on how much you have, how fast you eat it, what else you're eating, it can cause vomiting, dizziness, and of course, it can cause death. Cyanide is very toxic. It's about 1 milligram per kilogram body weight. So it's not very much. Just a couple of grains of sand of sodium cyanide is enough to kill me. You can get chronic conditions and you can get konzo. You get epidemics of tonzo, and these are related to environmental and social factors. Now, the processing methods basically all work on that system of bringing the beta-glucosidase into contact with the linamarin before you eat it, so the cyanide goes into the air. So whether you're grating it, that's what you're doing there, particularly if you mix it with water as well. Drying it itself doesn't do so much, but grating it up and then drying it and then wetting it will. Soaking will disrupt a lot of the cells, not completely. These pictures here of a woman in Mozambique who's pounding up the cassava flour. Now that's in itself is not going to release the cyanide, but once she's pounded it and mixes it with water, then the cyanide will be released into the air. And what we have here is a picture of a tipichi, which is, I've got a little model one, which is the standard one in South America. This is just a little toy one. I don't know if yeah, okay. So you put the stuff in there, you fill it up, and you stretch it, you put the slurry of the cassava in it, and you pull it tight, and the water runs out. And really about 95% of the cyanide is released in that way. And then, of course, there's the wetting method that was developed by Howard Bradbury. So on the right, we've got the test kits that Howard developed. And unfortunately, Howard has now passed away, and he's a great loss to the community. But Jan Elliott, I don't know if she's on the call, did these, and Howard also started the Cyanide in Diseases Network. Howard's, with others, developed the wetting method, where you grind up your flour, mix it with water, put it out in the sun to dry, all the water goes off, cyanide evolves, but it does have to be warmish, over 25 degrees, for the cyanide to go into the air. And that really removes all of it. So it doesn't have to be a problem at all. And I know many of you on the call know that it's not a problem because you eat it all the time. Now, as I said, I started working on Eucalyptus, which made cyanide. And as I read it, I kept coming across all these other things, like papers that Howard Bradbury had written, papers that Julie Cliff had written. And I think Julie Cliff has been very inspiring in this. And she was we had an earlier webinar from her of her discovery of that first discovery of the epidemic, together with Hans Rosling in Mozambique, where they investigated this disease that was Konzo. And it was definitely Konzo epidemics in Mozambique certainly coincide with high cyanide in droughted cassava. So is it because the cyanide, there's not enough water to wash the cassava, or do the plants actually make more? And later work, this is 30 years later, so Howard led a group, and here are the authors here, who they tested flour in the Mozambique markets. Bradbury never went there, so this work was done by others on the list here. But in a normal year, the flour in the markets in Maputo and places would be 15 to 20 parts per million cyanide. In a drought year, it's well over 200 parts per million. And the World Health Organization recommendation for food is a maximum of 10 parts per million. So you can see there is certainly an association with drought. And if you don't know where your food is coming from, that could be a concern. So this brings us now to the question of what are the environmental factors that might affect cyanogen levels? And will cassava be safe to consume under future emission scenarios? So on the left, we have a picture of a plant taking up light, carbon dioxide, and releasing oxygen. We know that driving carbon dioxide in the atmosphere has a direct effect on plants through the process of photosynthesis because it increases efficiencies. We also know there's the indirect effects through change in the climate, higher temperatures, rising sea levels, rainfall, climatic extremes. And so the question is, I've been working on other plants, but then more in the last 20 years, I've been trying to work out, will cassava be more or less toxic in the future, and how can we predict how that would be? So just by way of introduction to this, we did a small case study in Mozambique. This was with Anna Burns and Tim Cavagnaro from Monash Uni, with Anna Zacharias and Celeste Kawambe. And we collected cassava plants in Maputo, Nampula, and Lupo, and measured their yield, their nutritive value. I'm not talking about that today. We used this index, carbon isotope index, which is an index of water use efficiency and shows whether the plant's droughted or not. And we did 2 experiments. So in one of them, and this was a lot of work that other people did digging up. I wasn't even here at on this field trip, a huge amount of work, and I've just shown you one part of that experiment. Here's the total amount of cyanide in parts per million per dry weight. These were 4 different clones that were grown at Nampula and Lupo. And you can see that this is the Lupo sites, the 4 different clones. And you can see that you can get different concentrations. So they don't always, this 008 is, about the same at the 2 different sites, but you get a very get a very big difference between the cyanide of this particular clone at the two different sites. So the point I'm trying to make here is that because a variety might be sweet in one site, doesn't necessarily mean it'll be sweet in a different site. Sweet cassava meaning that it has less than 100 parts per million cyanide in it. So you can't necessarily extrapolate from one site to another. And it's important if you're going to be growing sweet cassavas, that you grow them in a number of different sites to know whether they're consistently sweet or not. And you can see here that 008, I don't know what it is, and this one are both consistently sweet. But the other ones, if you test it at this site, you might think, oh, that's sweet. But if you put it to the other site, then it's not. Now, the other one I've got here is a whole lot of different clones that were on Annabella Zacharias's site at just out of Maputo there. And we measured on the same plant, the tuber parenchyma, that's the starchy bit that you eat, the cortex, the outer part of the inside of the peel, and the leaves. And the point I want to make is that we've ordered these in increasing amounts of cyanide in the tuber parenchyma, the starchy bit. And you can see here, if we look at the leaves, there's very little relationship between leaf cyanide and tuber cyanide. So again, if you want to be testing for sweet cassava, you can't rely on what the leaves are telling you, because that may not be predictable. It may be for the clone in your region where you're growing it, that may be the case. But overall, you can't make that assumption. So there certainly needs to be some more work there, understanding why this is so. So I'm going to just show you some results of some simple experiments that I've done. I can see my time's getting away already. Now, these were two what we have honours students. We have like a bachelor's degree in Australia. We have an extra year that students can do where they do a small project. And you can see these plants, they only have 9 months to do these projects, so we don't have long. And you can see what rubbish the plants are compared to the stuff that you grow in the field. But these plants were very dry. These ones were droughted for 4 weeks and then rewatered. Sorry, droughted for 2 weeks and then rewatered for 2 weeks. These were droughted for 4 weeks, and these were watered for 4 weeks. And you can see the biomass, no big deal. Plants with water grow better. But what's interesting is that we found that the leaves and the tubers of the plants had very high levels of cyanide in the droughted conditions and low levels in the watered conditions. But interestingly, the rewatered plants had recovered and reduced. So these had been rewatered for 2 weeks. We also measured the leaves before and after we rewatered them. So we took, samples from individual leaves, and we found before they were rewatered, they were very, very high in cyanide. And then we sampled them 24 hours after they rewatered, and it was already down. So it was actually quite a quick response that decreased. So this is an interesting study, but not, the temperature they're growing in the light isn't anything like real conditions. But what it does do is, if we're looking about optimizing these resources, in this case, we've got reduced photosynthesis, reduced growth because the plants didn't have enough water, and therefore, we've also observed that the plants can reallocate those resources to defense, in this case, cyanogenic glucosides, and presumably making them more resistant to herbivores and pests. So just to increase the complexity a bit, and I'll just cover this very quickly, we grew cassava plants, another honours project, again, little tiny plants. How does this relate to the big plants in the field? Who knows? We grew them at 23 degrees. That's too cold. They really don't like it. Versus 34, and the same watering regime as the other experiment. And we see low temperature watered, low temperature droughted, high temperature watered, high temperature drought. So again, you water plants and they'll have more tubers. There's no big deal there. But also that they grew better at the higher temperature. But just getting now to the cyanide, we actually found that in the watered plants, they actually had less cyanide at the high temperature than the low temperature. And I think for these plants growing at 34 degrees, they were actually happier than when they were growing at the lower temperature. So in fact, for cassava, the low temperature was the stress, not the high temperature. But in the droughted plants, and we've got some other data on this which shows this difference greater, is that really higher temperatures exacerbate the drought. So if we're thinking of increasing in higher global temperatures, the drought, whether or not droughts themselves get worse, the plants themselves will experience more stress because the low water plus the high temperature makes the experience more difficult for the plant. And so in this way, growth is reduced and cyanide is increased. Now, just to what we've found for other plants, and I'm just going to flick back to this diagram because it shows it more clearly, where you have the watered plants have less and the droughted plants have more. Is it just because the plants are smaller, or do they actually make more? We have not demonstrated that for cassava, but for several other species, we have demonstrated that the plants actually make more of these compounds. They actually switch the pathway up under drought. So they actually become more cyanogenic, not just because the plant's smaller. It's not just a concentration effect. It is, in fact, an upregulation of the biosynthesis. Okay, so that's the last one I want to talk about here is the direct effect of carbon dioxide on cyanide production in cassava. And I have to give you a little bit of a plant physiology lesson because I know we have dietitians and neurologists and public health people on this call that probably don't know a lot about plant growth. But just in its simplest form, plants use carbon dioxide and water, the energy from sunlight, to make starch. The enzyme that catalyzes this reaction is really important. It's called rubisco, stands for ribulose bisphosphate carboxylase oxygenase, which I'm not going to ask you to remember. But that is the most abundant enzyme on the planet, and it makes up about 40% of soluble protein in leaves of plants is in this one enzyme. So it's an incredibly important enzyme. So you think if carbon dioxide goes up, starch will go up. So, carbon dioxide is plant food, but it's not quite straightforward. So you think, I'm just going to demonstrate this with my hands for a second. So maybe I should stop sharing. No, I can't be bothered. But if you think the plants should grow, increase in size by 40%, let's say, at high CO2, but actually they're only 25% bigger, not 40% bigger. So why is that? They're not making as much starch as you'd expect. And it's because this enzyme has two different functions. It catalyzes the fixation of carbon dioxide, which is this one we have here, turning carbon dioxide to starch, but it also catalyzes what is often referred to as a waste product. I'm not quite sure about that, but it's called photorespiration, and it combines with oxygen. And about 25% of the light energy that plants trap is wasted in this way. And so what happens is that when you give the plants more carbon dioxide, that favors this pathway. And so to grow the same amount, it doesn't need as much of that enzyme. So it can actually, that enzyme is expensive. It's a big enzyme. It has it's quite complex. It has a high level of nitrogen. If the plant, and it's a big investment because it's 40% of soluble protein in the leaves. If the plants only need to make, so it's 35% of soluble protein, that's a big saving. But what that means is the plants don't grow quite as much as you'd expect. And this has been shown for many different plants. And I showed this for these eucalyptus that I was working on. So if we look on the left here, so here's the concentration of this rubisco. You can see how old this data is. This is for rice. So if we had a certain amount at ambient carbon dioxide, then if it's 700 parts per million, the plants actually only have 65 or to 70% of that same amount of protein in the leaves. So you can see this. Then what happens is that this is in, say, let's talk about other crops. If there's less leaf protein, there's less available to go into the grain. So let's say we have barley. This is, say, just what you'd expect at ambient CO2. Okay, you have a certain amount of grain protein. If you grow barley at twice ambient carbon dioxide, you get a 15% decrease in grain protein. Rice, wheat, about a 10% decrease. Potatoes, about a 15% decrease. Soybean, not much decrease. This is probably because soybean is nitrogen fixing and it's not nitrogen limited, so it can make as much photosynthetic protein as it likes, and it doesn't affect it. So that's a bit of background. So plants downsize that protein machinery. And because that uses so much nitrogen, my question on my research for my PhD and ever since is if the circle is the amount of nitrogen in the plant, and let's say about 25% of the total nitrogen in the leaves is rubisco, the total nitrogen, not just the protein. And there's about this much as cyanide, let's say, for example. If when you grow them at high CO2 and they're only going to need like half the rubisco or maybe 40% less, do they turn that into cyanide or not? So that's the question. So it's not just about growth, it's actually how do plants use their nitrogen at high CO2. So let's think about cassava. So when I did that study and then I read about cassava, I got really frightened because we'd shown for many other plants, eucalyptus and so on, that in fact they do reallocate this cyanide. They reallocate this nitrogen and put more in cyanide, less in protein. Clover does it as well. So I'm thinking, oh my goodness, that means cassava is going to become a lot more toxic in the future. Turns out it's a little bit different in cassava. So in cassava, here's all the leaves doing their photosynthesis, transporting the photosynthesis, and then it's stored in the roots. And you have those big chunky roots that it has. So the questions are, if you have more carbon dioxide, do you just have more photosynthesis and growth? Or do you have reallocation because this photosynthesis is more efficient to defense, to the cyanide, and to longevity? Now, it turns out one of the things that signals from plants to go to downsize their system, so to go to the right rather than the left, is the buildup of carbohydrates in the leaves when they do photosynthesis. That signals to the plant, hang on, you're doing too much photosynthesis, it's too much. Of course, the plants are not thinking. You're doing too much, downsize the system and save some energy, don't make so much of this photosynthetic enzyme. But in something like cassava, you have these huge tuberous roots. They even just pack all, as you know, the carbohydrate just goes in and in and in, and you don't get the same signal. So the plants don't downsize in the same way. So here's the results just of one experiment we did as a free air CO2 experiment. That's what this stands for. Free air CO2 experiment. We did it in the field in Illinois. Yes, you can grow cassava in Illinois between May and October. And we showed, yes, you get over 6 months, you get, this is, at the time, this was the ambient atmospheric carbon dioxide. It's worth remembering that even already it's 420 outside my room right now. And then this was the high CO2, and we got lots and lots more tubers. They're only little. Mainly you get more tubers rather than the tubers being bigger. You get a greater number. Then the actual amount of cyanide, I'm just going to go back, but the actual amount, like cyanide per gram, linamarin per gram, ppm cyanide, is exactly the same in these two. But what we did find was in both the leaves and the tubers, the this is a way of expressing it, but essentially it's the cyanide to protein ratio. So at high CO2, you have much more cyanide relative to the protein. So again, there has to be people need another source of protein other than cassava, which is not high in protein. Certainly the tubers are not high in protein. So that you can make up for that, because you need protein to detoxify it. So we've repeated this experiment several times. The biomass, when you grow them in soil, they all get bigger. In all of them, leaves and tubers, you have less protein growing at elevated CO2. So you do actually get that less protein, but it doesn't result in any kind of allocation. One study showed an increase in cyanide per ppm. One showed less cyanide ppm, and two showed the same cyanide ppm. So I don't think the direct effect of carbon dioxide on cassava is the big issue. The big issue is the effect of climate change, increasing stress from high temperatures, excessively high temperatures. Obviously, cassava likes high temperatures, but the over 40 temperatures, and particularly how that combines with drought, because we know repeatedly that will increase cyanide and death. Now, see this diagram? Very complicated. Good news, I'm not going to take you through this diagram. I'm not going to go through it, but what it is, it just shows you there's lots and lots and lots of things that affect how much cyanide, much linamarin is in the plant. So how then do we deal with it? The question is, it's going to be very hard to predict what is done. So, and I took this from a paper by Jess Lyons, who some of you will know, and you met her in some of our discussions about cassava and Safe Cassava. And this is in her lab. She grew these plants up to being huge. This is just the seedlings. And here they are in tissue culture. This is the pathway to make linamarin and lodestralin in cassava. And what they did, her group, is that they made, using CRISPR-Cas9, they got rid of one enzyme, the other enzyme, and both enzymes. Because if you can knock out that first pathway, you will, in fact, knock out the cyanogenic glucosides altogether. So you have a completely non-cyanogenic, completely non-toxic plant. Now, she showed here's the results, which don't look very good. I'm hoping we can get Jess to give a talk on one of these webinars, because she's the only person that's really done this in such a beautiful way. Here's the wild type plant. Here's knocking out the D1. Knocking out the D2, but not the D1. This is the D1, but not the D2. And these are three different backgrounds that she's put it in. So you can see, and this is knocking out D1, but not D2. So that's much lesser. And this shows that D2 is dominant in biosynthesis. And if you knock out both of them, you can remove all the cyanide altogether. Now, her work showed that these plants grew exactly the same. Some older studies showed that they don't grow as well in early stages. I've done this in sorghum and found that it does have an effect in very young plants, but not in older plants. So I think there's some more work there that needs to be done. But, is this the answer? Who knows? It's certainly to have GM. Well, strictly, this is not GM, because it's actually just making these enzymes non-functional. It's not introducing any foreign DNA. So in Australia, anyway, this would not be classified as a genetically modified organism. So I've got about 3 slides. How do we make cassava safe now and into the future? You could not eat it. Well, that's stupid. 1 billion people eat it every day. Of course, people are going to be eating it, and people in South America have been eating it for 5,000 years. We know that that's ridiculous. But lifting people out of poverty and giving them a very diverse diet, particularly one that's high in sulfur amino acids, would make a very big difference.
do we want to make knockout cassava? So you could have naturally sweet varieties. You have to be careful about the environmental effects there. Maybe that's okay, but we do have to think about when you transfer them. There's a lot of complexity around genetically modified organisms. Would they be acceptable? These are, as I said, the CRISPR ones are technically not genetically modified because they do not contain foreign DNA. There are some question marks over the interaction between growth and removing the cyanide, but probably it is completely okay. But there's a lot of the world growing cassava, and I just cannot see the world growing transgenic cassava. So, which leaves processing as well as another important thing. And I think Howard's wetting method has been shown to be highly effective. Different parts of the world have different types of processing, you know, the fufu and the gari and all of those things have very different types of processing. And it may be that you only need to do high-level processing if there's a drought or at certain times of the year when consumption of fresh cassava is high. So that's finishing the science part. I now just want to introduce you to the International Safe Cassava Network, which is what this is and what you are all part of. So we initially started a working group for Safe Cassava, and this took many years, and it started in its current form in May 2023. So in 2011, together with Julie Cliff and Tim Cavagnaro, we had a workshop in Johannesburg with a lot of Mozambican and Zimbabwean colleagues, and we thought this is, to really have discussions about that, but we didn't get that far. In 2018, Felix Zelda, this man here, is a chemist who's got a new method for testing for cyanide. He invited me and Julie to come along and Joelle to have a discussion about cassava and safe cassava again. And then we thought, well, we really should do more of this. And then 2019, you can see we've got a few more people. But then COVID hit and nothing happened. But while COVID was on, Julie Cliff, who many of you are familiar with, has done so much groundbreaking work on Konzo, was in Australia. And Jan Elliott is also in Australia. She's in Canberra. And we got together and thought, we think we can actually do something a bit more with this. Let's try and work out something, take it further. And so this has developed further. And so here's a more recent photo of Julie, same cafe, I might add. Julie, me, and Kira Ma. So you might be aware of Kira, who's the one that sends the emails to people on the mailing list. And I just want to, so part of this, initially, we have just started with a steering group or a working group that was people that we knew and had invited. I think there's about, Kira will know the exact number, she might put it in the chat, about 30 people involved in that. And we probably have about, 10 people every couple of months to discuss some of the big policy issues and to hear some of the latest research that perhaps is not ready to go into the journals and not ready for a recorded seminar such as this one. I also just want to acknowledge here that the earlier workshops were funded either by AusAid or the Australian Centre for International Agricultural Research. But currently we have now got money from the Australian government, again, the Department of Foreign Affairs and Trade, and which is what AusAid has turned into, and it's been funded through the Australian National University. So I just want to thank the ANU for that, and also part of the money for this. I'll come to in a minute. The next thing. Okay, so you can see here we've got a few of the people who are online tonight, including Kira, who, because it's very late in Melbourne compared to the time in Europe or in the US, where it's probably very early. So we've been talking about the real need to have a symposium, and I wanted to get everybody together in a room. but it's not realistic, right? The cost of getting everybody together, the carbon emissions, it's not appropriate. So we have now started this, expanded this, the working group into the Safe Cassava network. And if you got emails from it, this was based on the original email list from the CCDN that Howard Bradbury started. And so we've called ourselves the ISCN. And I think, Kira, to tell you, we were just in the process of buying the domain names for this, with a view to putting a website together, or if we need to. But what we decided to do is plan an online symposium. Enormous thanks to the Global Plant Council again, who have offered to host this, and Isabel in particular. And we'll hold it over 2 days. We'll have 4 sessions, 2 sessions on one day, 2 sessions on the other. As Isabel is in Spain, all sessions will be at a sensible hour if you live in Europe and probably most of Africa. If you're in America or Australia or in India or China, we're going to have some hard hours to settle in. And the potential topics are that we could have something on cassava, different people talking about growth and so on, and the transformation methods, and conzo. And we'll have a session on public health. And then the last one will be on food systems and the food environment. And we'll have 2 chairs for each session. So far, we just have Umberto and Torkild for taking the lead on these 2, but hopefully we'll have some early career researchers that will be able to assist them. So more information will be available through the Conzo Prevention Unit through the Australian National University. And they will be supplying some funds to enable us to employ Kira Ma again as the secretariat for organising this. So we haven't decided yet on these 2 dates. If there's anything that really clearly clashes with something in your country, please send Kira an email or put it in the chat now, and we'll take a note of it. The QR code there will take you to the Conzo Prevention Unit webpage, and we will have a page on that. So we'll make a page for ours. We have permission from the ANU, from the Australian National University, to do that. So finally, I just want to thank 2 big people I want to thank. I have thanked others that we're through. Howard Bradbury, who of course has now passed away, who was just the nicest person and really got me into this in many ways and introduced me to Julie Cliff and set in train a lot of things. And Isabel Mendoza from the Global Plant Council, who's hosting this. And also I want to make special mention of Kira and all of other people who have actually done the work and the people who paid for it. So thank you very much for your attention, and we'll open it up now for Q&A.
So thank you. Thank you, Ros. Thank you for that last slide. Let's go dive into the Q&A. We have lots of questions coming in. Just a reminder to use the Q&A tool for sending your questions, because we are not going to be able to manage all the chatter in the chat. So this is the clear way to get your questions out. So we will start.
I just had one.
Yes.
I just had one thing from the chat. I just saw about the certificate of attendance. I think you can, if you go to participants and see the list, can the people in the webinar see the participants?
Yes, yes, I can.
Yes, so you could go to that and just do a screen snap, and that will show that you're logged on if you need that for your employer. Sorry. Okay, Isabel. I know I talked too long. I apologise. I did have a health emergency, but I had a health emergency for a family member today, so it didn't quite finish my preparation in time.
No, I think it was perfect. We have still half an hour if we need it. So let's go with the first question. And it's less about your specific talk and more about the Safe Cassava network from Hafiz Bashir Hamad. And he's asking how can anyone get cassava seeds? Is the Safe Cassava network able to help on that? They are
No, the
contacting us
network,
from Pakistan.
yes. So thank you. I don't know. I think hopefully someone else in this network will know how you can get to it. I would have thought you could get it in Pakistan. It's certainly grown in countries nearby. So I know that they do grow some in India. So I don't know whether it would be too difficult, but it must be there. If anyone can help, please send a message to Hafiz in the chat.
So next question, I will go by order of time that we receive these questions. So is it possible to use cassava as a fodder for feeding livestock?
Yeah, that's done quite a lot. And people often use the peel to feed, because the peel is very toxic. They feed that to their animals, but you do have to be careful about that. So there's quite a bit of literature about how to go about doing that. Again, other people who are on this call will be far more expert at that than me, but you can. But you have the same issues as with humans. If you have too much cyanide and not enough other food, you can get toxic effects, but different animals can tolerate different amounts. So cattle are much more tolerant of high ppm, for example, than humans.
Thank you. So there is a next question. I see some people raise their hands, but we are not giving we are not going to open the microphone. So please send your questions through the Q&A tool. Next question for Mohammed Kuranakaran. I'm sorry if I butchered your name. How can we assess the cyanogen content at farm level? Is there an easy way, if it exists anyway?
The kits that Howard developed, that I showed the picture of, that Jane Elliott makes in Canberra, she does that as a volunteer. She sells them to people who can afford them and gives them to people who can't. They're great. They're fantastic. But I mean, this is a real issue, and I think it needs solving. And we've got, I don't know how many people on this call, but there's got to be someone who can take this and make a commercial property out of this so that people can test their cassava on their own at their farm level. It needs to be done. At the moment, it's not easy.
Mm-hmm. So we have another question from Eugenia Pitulauditi. Again, sorry for budgeting your names. Can cassava plant grow in the Mediterranean climate? How we can find those plants?
It will certainly grow in a Mediterranean climate, but not. So, for example, Melbourne is, what are we, 37 degrees south? So 37 degrees north, what's that, kind of middle of Italy? It's too cold. So you can't grow it outside. You can only grow it in summer. So it doesn't really grow below about 20 degrees and 15, maybe down to about 18 or 19 degrees Celsius. But so it won't grow in winter, but you could grow it in the summer. How do you find plants? No idea.
Maybe in the seed banks. There's some seed banks, not in all.
Yeah, I'm sure there would be in different countries, there would be people who had access to it. So, Eugenia, if you want to put in what country you're from, there may be someone else on the call who may be able to. Yeah, so Torquil's just commented that it doesn't grow in South Africa. Yeah, because it doesn't tolerate frost. Absolutely. So you're from Greece? Yeah, I would have thought you'd be able to get it there, but I don't know anybody who's growing it there. But it would grow well in Greece, except it's not going to survive in the winter. You have to start again each spring.
Then we have a short message from Matthew Bramber, who says, "Hi, Ros."
Hi, Matt. Good to see you.
Then we have a question from Polycarp Okafor. It's quite a long one, so I'm going to read it. What concentration of cyanide or cyanogenic glucoside exposure to humans can cause cancer as seen in Mozambique? I'm asking because we in Nigeria consume lots of cassava products, but you can hardly see people with cancer. This assertion I confirm from my works on metabolism of cassava cyanide.
Yeah, I don't have the expertise to answer that. I would have thought it's probably something to do with it being in a mixed diet. Is it possible to unmute Torkild? Just look score.
Who?
Just look for the Swedish name. Torkild. THORKILD TYLLESK. TT. It's on my
Torkild?
list.
Yes.
Yes. If we could unmute that. Torkild, do you know the answer to that question?
Torkild, we are putting you on blast.
We're unmuting you so you can answer. April will come back. I mean, the short answer is there are some different techniques in Nigeria, but I suspect it's because generally it's eaten as part of a mixed diet. Maybe it's not so dry. There's different things. I don't know. I really don't know, and that's why we need the symposium.
Okay.
Yeah, let's just move on.
So from Luigi Guarino, is there a lot of diversity in cyanogenic content among sweet cassava varieties?
Again, I'm not an expert on this. Of the ones that we've measured, yes, there is. But if you read the paper by, was on one of my slides, Bredesen, maybe Kira, if you could find that reference and put it in. We did a lot of genomic sequencing, big international consortium, and that shows that some of the varieties that we thought, the sweet ones we thought were really different, are actually really similar and that sort of thing. So that will give you a sense of the diversity of what's been sequenced. There've been more papers done since then, but that's a good one.
We will try to paste this reference in the chat.
There it is.
Yeah,
Thanks,
there
Kira.
it is. A question by Matthew Bramble. What are your thoughts, Ros, on how climate change will affect other crops and cause our reliance on cassava, particularly in regions where cassava is not as common?
My observation, and as I feel like I'm not an expert, I look at all these people on this list of attendees, and I know that you know so much more about it than I do. But we know that the plants are likely to become more toxic with drought and extreme conditions. Certainly if you have saline water, they will. We also know that people may become more reliant on cassava because it's the only thing that will grow. One of the issues seems to me that the spread of cassava into regions where it has not been as common must be accompanied by the knowledge of processing. So it seems to me that one of the problems is that cassava is growing more and more into different areas. But as it's planted in those areas, people don't know about processing. I mean, it's grown in Australia now. Australians have no idea that they shouldn't just eat raw cassava. So I think that is a very important point. And the great need for education along with this for the processing. Before you go on to the next question, I noticed Umberto has said in Mozambique, the trigger for Conzo is the consumption of bitter cassava. So I think that's right. So plus a low protein diet. And I think bitter cassava, we know that some cultivars are more bitter. That's a start, but we also know that drought can make the plant more bitter, accompanied with a low protein diet. That's the key.
Yeah, Thorkild also pasted here an answer to the question.
Yes. Okay. So you have to have a very high cyanide diet. And I remember when Howard Bradbury was doing some surveys with, I think it was with Benin in Congo, and they were asking asking people how often they ate cassava, and it was the wrong question, because if they ate cassava once a day, it meant they were having 1 meal a day. If they ate cassava twice a day, they were having 2 meals a day. And if they ate cassava 3 times a day, they were having 3 meals a day. In other words, people were only eating that. So if you can mix the diet, you won't get Conzo. And also, to some extent, you do build up a bit of tolerance. I mean, Torquil will know more than this, but the enzyme that you break down the rhodonese, people that have a little bit of cyanide in their diet will make that a bit more readily than someone like me that doesn't eat cyanide at all.
So should we move to the next question?
Yes. Yep.
Next is by X. A Glitter. Would diminished cyanogenic glucoside production make the crop more vulnerable to disease and pests?
This is a very interesting question, and the answer is to make it zero probably would make them more vulnerable to pests. Less disease. It seems to be these compounds aren't particularly effective against disease, but they are effective against pests. We've certainly got some zero cyanide sorghums, which don't seem to be affected by pests at all, but then all the other paddocks are full of cyanide. So we don't know quite what's causing that. What I am more concerned about, and I didn't talk about this today, is that it does seem to be some crossover between bitterness and the ability to tolerate stress. So it seems to help the plant tolerate stress and manage the nitrogen in the plant. So we do have to get rid of it completely, may not be the best option in the plant. If you could do as Jess has shown, you could knock out just one of the enzymes and the other one's still there, and so you're getting lower levels. That could be better. So you get a guaranteed sweet variety. Now, Kira's been trying to replicate some of these experiments for your honours project, where we had some varieties and stressed them and rewatered them and so on. And it's quite hard to do under controlled conditions. And the other problem is that we don't actually have any bitter varieties in Australia, really bitter ones in Australia. So we haven't done it. But some of the work we did in Fiji would suggest not so much the lack of resistance to pests, but the ability to tolerate drought. And of course, there is all that work that Lindley, I can't pronounce her surname, did in Malawi, talking about the acceptance of bitter varieties, which give women control over the food source. And there's a lot of social things around people wanting to grow bitter varieties. If that is the case and people grow bitter varieties, you do have to make sure you process it. And in Southeast Asia, they grow bitter varieties because it's mostly for industrial starch, but then people do eat it as well. So it could be potentially a problem there.
So we have next a question by Charles Ologidi. The problem, even with the use of CRISPR, is acceptance. There is also perceived biosafety concerns. Do you have any comments on that?
Yeah, I've got lots of comments on that, and certainly one of the issues that the Global Plant Council has talked with, and one of our research papers deals with this type of thing. So yeah, it is an issue. Some countries are very happy to have all kinds of genetically modified organisms. I mean, those wonderful pest-resistant cowpea they've introduced to. I'm just trying to think, is it Nigeria or is it Niger? Or yeah, I can't remember. But I mean, that's brilliant stuff. So yeah, it's a difficult problem. Personally, I don't see any problems, but there is perceived concerns.
Yeah, there are a couple of reviews on these regulatory frameworks around the world that cover all crops produced by GMO techniques or CRISPR that have to be republished every now and then or redone every now and then when the policies change. But yes, there is a lot of difference regarding policy and also regarding acceptance. And yeah, that's a big issue. Maybe for another webinar to to devote the whole time.
Yeah, I think so. We can have another webinar on that.
Yeah, we have another question by Elena Castillo. That first, thanks you very much for your content. And do you know if there are wild relatives of cassava that behave differently under drought, producing less cyanide perhaps, or are there studies on wild relatives that you know?
There is been some work on the genetics of those that's been done. Alex Abono did a lot of stuff on that, and particularly in Amazonia and in Brazil, looking at genotype differences. Some very nice papers they've published. And it does appear that Manhot esculenta is in fact an ancient hybrid with another, what would be regarded as a wild relative. So there has been a hybridization event that probably made it more toxic. So it seems to be that a lot of crop plants have these cyanogenic compounds in them, and we think it could be something to do with when plants have been domesticated, there's a need to sort of manage the nitrogen in the soil. The plants can take it up, store it, and then use it later, or something like. We're not fully understanding, but the fact that 5% of wild plants are cyanogenic and about 40% of crop plants, even only if they have small amounts, that's too big to be by chance. So it seems that humans have actually favoured doing this. It's just cassava tips it over to being it can potentially kill you if you have the certain varieties that aren't processed. So I think more work on the wild relatives would be terrific. The tree cassava, which the Gladosevicii, I haven't said it correctly. That's clearly been transgressed into Cassava esculenta in Africa, and it's in a lot of the breeding stocks. That probably again is more bitter.
We have a next question from Ochanda Gola. How do you process cassava leaves to reduce cyanide content? There are regions where cassava leaves are eaten as leafy vegetables, and that could be a problem.
Yeah, it could be a problem. It's actually a very good source of protein as well. So it's a balance with that. I think again there'll be people in the who are on this call who would know better than me. But the principle is that if you bring the cyanogenic glucoside into contact with that enzyme, then the cyanide will go into the air. So I think one website I was looking at, they said, you boil the leaves, then you pound them, and then you eat them. And you think, well, boiling it doesn't kill would that not kill the enzyme? This beta-glucosidase enzyme is very, very stable, and it can survive boiling. So actually boiling before you pound will work. I always thought you had to boil first, had to pound first, but you don't, because that enzyme is so stable. I know in Mozambique they make a kind of a fermented pesto out of it, which they like, and I don't. But I'm not used to it. But yeah, it can be a great source of protein. But remember, it's low in methionine, so you do need other sorts of protein as well.
So next question by Geeks A Kletter. In your opinion, would farmers and households benefit from quick, easy, and cheap tests for cyanide and cyanogenic glycoside content?
Yes, I think that this is a great need. I think it would be terrific. I think, you could have something in every village or town or something. Not every individual would need to do it. You could even just have, sentinel households that tested to say, okay, it's getting dry, the levels are going up. Well, we'll need to switch from our normal just boiling and cooking. We need to use the wetting method or something that's much more efficient. Just a quick answer, Lindley, and I'm sorry, Lindley, for not even attempting to say your surname, Chawona Carlton. It refers there to her work on the preference for bitter varieties.
Yes, yeah, you mean in the chat box?
In the chat box, yeah.
Yeah. In fact, there is the next question. It's from Okon, and they ask, how can one join this working group for Safe Cassava? I'm a Nigerian, and cassava is one of the most consumed food crops in southern Nigeria. In fact, they add something on the chat that if anyone in Nigeria is willing to go into cassava research, we should collaborate, and adds their email address.
Yeah, no, I think that this is terrific. And the working group, the way it is now, to me, I see as just a start, just because they're just people that just happen to be there. If you email Kira and give a bit of background of what's going on, we can have that. I think there are a couple of people from Nigeria in the group already. Because they're at strange times, not everyone can come. But what I'd really like to see is at this symposium, if we could have a bit of time as a result of that to work out how we'd like this group to go forward. So this group that we're in right now, which is the International Safe Cassava Network. So I think we have the network, and then maybe the working group becomes like the organising committee for the network. Whereas the real business will happen in the network. But it's definitely, we need to really think about these things. We're at early stages now, and I think the fact that we've had so many people jump in on this call shows that there's a real need for it.
Jump in and staying.
Yes, thank you.
Yeah, we can discuss that later, but I think during the symposium we could organize some session just purely for networking.
Yeah, that's a good idea.
So we have another question from Samuel Mapesa Niawanga. Sorry for budgeting again a name. And they ask, increasing CO2 or the level of the cyanide correlate to the tuber size of cassava? Do you know?
So there's two different things that happen if a plant is bigger. It's like or smaller. So do they have the same amount of cyanide? And is the tuber bigger? Does it correlate? Not directly, no. So just you increase the CO2, you will get bigger tubers, and the ppm, the concentration of the cyanide will be about the same. So there'll be overall, if you took all the cyanide in the tuber, it would be more cyanide, but it's the same concentration. Mostly, we've done this experiment several times, and it's confusing.
Okay. So we have another question from Charles Oligidi. There are a number of crops like taro having anti-nutrients and could benefit from knockout using CRISPR. However, acceptance of products from the technology seems to be an issue in Africa. This is more like a comment than a question.
Yeah.
And
Yeah, I mean that's something that needs to be discussed. My group also work on taro, so I understand what you're talking about.
yeah, then there is contribution because it's not a question, by Luigi Guarino, with the link to the cassava gene banks around the world. It's available there. I'm gonna.
Oh, that's fantastic. Thank you for that, Luigi. That's really, really helpful. Whereabouts are you based, Luigi? Do you need to pop it in the
So
chat?
I'm gonna post this in the chat box just in case for everyone to see. And whenever we publish this on YouTube, I will add these useful links also to the box, to the caption of the video. So Luigi Guarino, if you can share with us where are you connecting from, that would be great. Yeah, so we have a new.
Oh, I see. I now see what Matt is asking in this question. So I'm going back to that question from Matt Bramble.
Yes.
So he's talking about how will climate change affect all the other crops and so there's two things there. One is because so many of the other crops will not grow as well because it'll be hotter and drier, there will be more reliance on cassava. That's true. Also, those other crops. So say, for example, eating wheat or rice, particularly those sort of grain crops, they will have less protein in them. So you won't get as much protein, or any leafy vegetable, and many fruits will have less protein in them. Some of the crops like maize and sorghum have a different type of photosynthesis. We've done a lot of work on sorghum, and there's a lot known about how maize responds, and they do not see this decrease in protein. So if people are still able to access sorghum, which is much more drought tolerant than maize, sorghum also contains these cyanogens, but not in the seeds. So you have to be concerned for feeding it to your cattle, but not the seeds. But yeah, if it won't have such a big effect on maize, and so except that of course the rainfall will be less, and so you won't be able to grow as much maize. So that becomes a problem. I hope that answered your question. It's just be a lot better if we just reduced carbon dioxide in the atmosphere and avoided the problem.
Perfect. Then we have two comments from Esperance Casala Advance, who was our speaker last time, answering the question about why conso was not so usual in Nigeria. It has to do with the acidity of the soil, she says, in addition to diet, as we commented before. So
Good, thank you for that.
thank you, Esperance. Then Percy Nimako Yamoa, it's also contributing to answer that question about why Nigeria seem to be different.
Yep.
And he answers, Nigerian consume adequate amounts of animal products alongside their cassava diets, which I believe helps to break down the cyanide build-ups, reducing the occurrence of conso or cyanide toxicity. Yeah.
Yeah, and I think that's also I think, Lindley, it was some of your work that you did around Lake Malawi, where if people were near the lake and eating fish, then you don't ever see conso. Fish are particularly high in sulfur amino acids.
So more or less in 4 minutes we will end. And I think we are not going to be able to finish all the questions, or maybe we can. What do you think, Ros? How you feeling?
I'll have a flick through and see what we can do. So yes, Charles Oligodi is talking about the relationship to goiter and cassava. I think that's something to do with the sulfur being used to detoxify the cyanide rather than going into the thyroid. And how does climate change specifically affect cyanogenic glucoside levels? Really, if you go to extreme temperatures, more salt in the soil, and particularly drought, you'll get higher cyanogenic glucoside levels in cassava. So if that is the case, and people may rely on it more because their other crops are failing, then we need to make sure for nutritional quality and safety, there needs to be an increased reliance on education so that the food is processed and safe. And you can make cassava completely safe using the wetting method or any of these other, some of these other methods. Some of them are more effective than others.
Mm-hmm.
So I think that's probably everything.
Okay.
So I've just sort of answered those.
Okay, great.
Yeah.
So I think we are going to wrap up. Thank you, Ros, and thank you, Kira, and thank you everyone in the call who stayed until the very end. As Ros already has shared, we are organizing a symposium for October. We will keep you updated of the specific date and the specific time slots and how to join. Then I also wanted to share with you that we are already organizing the next webinar for April, again on climate change issues, this time with Professor Danielle Way from the Australian National University. I've shared the registration form in the chat, but I will also share it again with you along with the link to the YouTube recording for you to rewatch this whenever you need. As always, you can reach the Global Plan Council via my email. Any other email that you find in the website is also functioning, and of course through social media as always. And thank you so much for being here. See you next month, and see you, Ros and Kira, soon for organizing.
Yeah, good. Yeah, thank you. I just made a note that webinar, of course, is the next Global Plant Council webinar and not particularly about cassava, but it will give you insights about climate change. Good.
Yeah.
So thank you very much for your attention, and it's great to see everybody. Good.
Bye.
See you, Poasa.
Bye, everyone.
Bye.